Hi, class. I'm sorry I can't be with you today. My sons are not feeling that well, so I've got to stay home and take care of them. But instead of just having a like homework day, we are going to try and get ahead on some of the material in this unit so that we can stay on schedule to finish what we need to finish. With that in mind, today we are going to learn about DNA replication. I'm just going to talk about it with you for a few minutes and give you some notes. The substitute should have passed out a notes sheet for you. I'd like you to listen to this video and take notes as you listen. Tomorrow we can go over any questions you've got before we do our lab related to DNA replication. It's okay to have questions about this. If you're feeling a little frustrated that you're not understanding everything, that's okay. Try your best. Look at the text I gave you, and tomorrow we will go over what you uh, have listened to today. Last week, we started off looking at DNA structure and history. This week, we're going to look at DNA replication and how DNA goes from one copy to two copies, or how our cell accomplishes that job. We will finish this material by Wednesday, so you can expect me to check in your homework on Thursday, and you should also expect a quiz to be due by Wednesday night or Thursday morning. That's when we will start on the next topic here. So you have a few days left, or maybe 1 or 2 days left to finish your homework. I would make sure you get that done today if you have some extra time. The other thing you're going to do today after you listen to this video is that you're going to get back together with your group or your plant group. You'll get your plants from the shelving unit in the corner of the room, and you'll fill out your next page of the phenology journal. I'm looking to take notes this week on Tuesday and Friday on that plant journal. So make sure you don't forget to get your plants out after today's lecture and look at those so you can fill out another page. Okay? Today we are going to learn about DNA replication. And it's really important that we always think about, why is our cell doing these things? What is the big picture behind all of this material I'm learning about? Today, DNA replication we're gonna go over basically how your cells copy all the DNA in your cell and make one copy turn into two copies. The reason behind why this occurs is because your cells need to, copy themselves and replicate. Your body's made out of a lot of different kinds of cells. All of those cells came from the replication process. How is it possible that all the cells in your body have the same copy of DNA in them? Well, DNA is replicated in every cell and then passed on to the new cells. And so today we're going to look at how that occurs and go over some details. The three major actions that occur when it comes to DNA replication are: one, the binding of enzymes. Excuse me. Enzymes really assist in doing almost every action that is required to turn one DNA molecule into two. Those enzymes we'll go over in detail in a little bit. The unwinding of the double helix is also going to be very important to this process. Their DNA double helix is wound up into a tight little ball, and your cell needs to unwind that to get at every single strand and make a new copy of it. The synthesis of the new matching strand is going to be the end goal of replication. Synthesis stands for to make. So we are going to make a new matching strand based on the old template strand that's in the cell already. For future reference, as we go throughout this talk, parental strands are part of the original double helix molecule. So when you think about a double helix, which is two sides of a DNA molecule, we call each of those sides a parent strand because it was already there. It was present before replication started. The name for the new strands are always called daughter strands. Um, those daughter strands represent what the cell is making in terms of making a new copy of DNA. So original strands are called parent strands, new strands are called daughter strands. This whole entire process we're going to talk about is referred to as semi-conservative replication, where one molecule of DNA is unzipped and made into two or replicated into two copies. As you can see with these new copies down here, one half of this molecule contains a blue strand, and that blue strand came from this original DNA double helix. So as it unwound, it's used as a template or a backbone to build the new strand off of. The new strand is initially in individual A, T, C's, G, and G's, but they are assembled in the correct order based on the old strand so that we can get two brand new copies or two new copies of DNA based on the original model. This type of replication is called semi-conservative replication because the new strand contains one old and one new strand. Um, all of your cells go through semi-conservative replication. All right, now we're going to learn about this process in 3 steps. DNA replication essentially occurs in those 3 steps. The first step is going to be to separate the parent strands. Separating those is going to require an enzyme called helicase. Helicase unwinds and unzips the DNA molecule. If you're confused as to what this might look like, find or picture yourself wearing a sweatshirt or something with a zipper and unzipping it with your fingers. Unzipping a zipper is exactly what helicase is doing. As soon as helicase is done unzipping the DNA synthesis starts to occur. DNA synthesis, remember, is the making of a new DNA molecule. That's going to occur with the use of something called RNA primase. RNA primase is going to create a primer, and primers are required for DNA synthesis to get started. As soon as a primer is laid down, an enzyme called DNA polymerase will start making the new strand at the end of the primer sequence. That primer sequence lets DNA polymerase know where to start, and then polymerase starts making the new DNA molecule after the primer. The combination of DNA and proteins called a replisome. I will be honest, I'm not going to refer to this vocabulary word a lot, but that is the name that is referred to when we talk about how DNA and proteins are helping to make a new DNA molecule. Let's watch this happen, albeit in the beginning of this, but let's just watch this happen initially based on what we've talked about. Let me go back and start again. Here's my DNA molecule. It's been unzipped by helicase. Primase has come and laid down a primer. That primer allows DNA polymerase to land on the strand and start building a new DNA molecule in the 5-3 direction. This molecule is built one nucleotide at a time towards the opening of this strand. For the time being, please ignore the top strand. This top strand is a little more complicated. So, just to review again, RNA primase lays down an RNA molecule. That primer gets the strand started. Polymerase lands on the primer and starts building a new DNA molecule. All right, so that's basically part one. Unzip the molecule, primase lays down a primer, and polymerase starts building the new DNA molecule. It might be important to add to this note right here that DNA polymerase can only make new DNA molecules in the 5-3 direction. That's based on the orientation of the molecule. We did spend some time last week talking about that, so refer back to your notes from last week if you have questions about that. All right, part two, the making of the new DNA strands. This is done by the enzyme DNA polymerase. Polymerase adds nucleotides in the 5 to 3 direction, or the end of the three end of each nucleotide. Um, I kind of think of this as it's easier to understand or memorize that DNA polymerase goes in the 5 to 3 direction. The sentence is okay, but I might write that down instead. The two functions of polymerase is to add nucleotides to the existing molecule and make that strand in the 5 to 3 direction. While polymerase moves in the 3 to 5 direction, DNA and RNA, for that matter, are always made in the 5 to 3 direction. This is the most important number orientation to understand. All right, why is that number orientation important? Well, if you take a look at this molecule again and ignore the animation for a second, you'll notice that the double helix is oriented in opposite directions. That original blue molecule is 5 to 3 over here and 3 to 5 below. Because that is the case, you cannot copy the DNA strand in the same way on either side of this molecule. If you go 5 to 3 right to left, or left to, I'm sorry, this is left to right for one strand, you'll have to go backwards on the other strand because each strand is in opposite directions. So where do we start with that then? Well, we start here on number 4 with the leading strand. Hallmarks of the leading strand are that it's made continuously, so no breakage occurs in the making of the strand. It is made continuously from beginning to end. And it is made, while it is made in the 5 to 3 direction, polymerase is moving in the 3 to 5 direction. Again, do not focus on this. I wish I could X this out. Don't focus on this as much. Focus on the synthesized continuously in the 5 to 3 direction, the 5 to 3 direction. So let's watch that happen. The leading strand is actually going to be best represented on the bottom. So watch the bottom. Primase comes by, lays down a primer. Polymerase comes by next, lays down our next nucleotide, and the molecule is built in the 5 to 3 direction towards the opening where helicase is unzipping this molecule. Again, ignore the top section for now. These little blue arrows represent A's, T's, C's, and G's. This molecule will be continuously built until the entire strand is opened up. Now we can focus on the top strand and go back to that animation in a second. Top strand is called the lagging strand. What are the hallmarks of this strand? Well, it's made discontinuously. That means with breaks. So the enzymes will have to stop and start, and this creates breaks in the molecule that have to be basically bonded together at the end of this process. So the lagging strand is made discontinuously with breaks. This creates short DNA fragments that have to be connected together. These fragments are called Okazaki fragments after the person that discovered them. Let's figure this out and see how it is different. Now notice the primer here is being laid on the leading strand in the 5 to 3 direction. Can't lay a primer down over here in that direction because the strands have to be anti-parallel or backwards. So primer gets laid down in the 5 to 3 direction going right to left. Polymerase builds a strand and that makes a first fragment. Primer goes backwards, lays down another primer, and then polymerase can lay down more nucleotides, making a new fragment. Eventually, these RNA primers have to be removed. So polymerase will remove them and add more nucleotides. And then an enzyme called ligase seals and bonds all those fragments together. In the end, the leading strand and the lagging strand should not look different from one another. It is only different in how they are made. One is made in pieces and has to be sewn or bonded together. The other is made continuously and does not need to be bonded together after the fact. Let's watch that one more time. Primase on the leading strand is laid down in the 5-3 direction. Polymerase adds nucleotides, A, T, C's, and G's in the 5-3 direction. Primase lays down a primer in the opposite direction on the lagging strand, allowing DNA polymerase to make our first Okazaki fragment. A second is made while the strand continues to open up. You would assume a third would be made right here as the animation, if the animation were to continue. Primers are removed. Primers like these would also have to be removed as well. And ligase would seal the connection between the two fragments. All right, that is it. We have one last part to go. Okay, here's a quick animation or a quick visual for this process. I'm going to link in here a great video that will show us this process again and get you to review. Lastly, part 3, stitching the pieces together. The enzyme known as DNA ligase is an enzyme that, as this process wraps up, will help connect all those Okazaki fragments together. So ligase in the replication process will connect all the fragments together so that the entire DNA strand is one continuous DNA molecule with no breaks. The molecule, remember, will be anti-parallel. This is why you have to copy the strands in different directions. There's another visual for this process as well. I think that's pretty much it. That wraps up DNA replication. So with that being said, I would check your notes, make sure you have all those down. You could go back and pause where you need to so you can finish writing. I'm going to link in here a YouTube video that will help you visualize the process once again. What I would recommend you do at this point is finish up doing your homework and the vocab journal. Those will be checked in on Thursday. On Wednesday, I'll review this with you and we will do a lab where you build a 3D model of this so that you can actually see the replication process happening in action. Okay? As this video finishes today, don't forget that you have to check on your plants, fill out one page of your phenology journal, and get going on that as well. So you've got a few things left to do. After you finish this video, watch the other YouTube video I've linked in here. That's step 1. Step 2 would be to do your phenology journal, get your plant, meet with your group, and do your journal entry for the day. And lastly, your third piece should be to finish up your homework and that vocab journal I gave you on DNA and replication. That stuff will be quizzed on in about a day or two. And if you have questions, it will be a good idea to get all those questions to me tomorrow so that we can go over them as you prepare for the quiz. I hope you have a great day, and I look forward to seeing you tomorrow. Bye.